So, good morning. Welcome to this progress report. This is a little bit, it's not a regular progress report. This is more like a mixture between a lecture and a progress report. And it's about energy metabolism in the retina. Some of you may have heard some of it already. I know for my people should be familiar with this. But perhaps a couple of details that may still be interesting and new. And I hope that for the rest of you, it will be interesting. Okay, thank you for adjusting the camera. So let's get started. The topic for today is the energy metabolism, and this is kind of a two-part little mini lecture series. Next week there will be an update. Today, this is what I'm really presenting today was basically published in a paper in eLife last year. And next week, that will be the new stuff that is unpublished as of yet. So what I'll be talking about, there will be a brief recap about general biochemistry, glycolysis, Paquet-Cycle, oxidative phosphorylation, these kind of things. And then I will talk about metabolic coupling, what that is. A little bit about the evolution of energy metabolism, different effects that are related to this, and then I will introduce our experimental system, experimental retinal electron cultures, and how we go about working with these and analyzing this. I will talk about different biochemical cycles, Cori cycle, Cahill cycle, and Krebs-Cycle, Mini-Krebs-Cycle, and in the end, I will talk a little bit about how the energy production relates to diseases of the retina. Okay, so I don't need to introduce the retina to you. We all know that there's basically three main cellular layers: the neuroretina, the outer nuclear layer, the inner nuclear layer, and the ganglion cell layer. The outer nuclear layer harbors the photoreceptors. There's rods, cone photoreceptors. And we probably also all know that photoreceptors have a very high energy demand. And this energy demand is also kind of mirrored by a very high perfusion rate in the retinal vasculature. The choroidal vasculature has the highest perfusion rate anywhere in the body. And what is a little bit interesting also is that the retina is shielded from the rest of the body, from the blood circulation by the blood-retinal barrier. The energy consumption is pretty high, or very high, probably the highest in photoreceptors of any cell in the body. And this is, at least in part, attributed to the dark current of photoreceptors. And now, of course, when we have this very high energy consumption, which is probably so high that it reaches the biologically possible limits, this may also be related to the vulnerability of photoreceptors that we see in certain diseases. Okay, so now I would like to have a brief recap of some general biochemistry, glycolysis and Krebs-Cycle. Glycolysis, just very briefly, splits glucose, a 6-carbon molecule, into 2 pyruvate molecules, each one having 3 carbon atoms, and that liberates a little bit of energy in the form of 2 ATP molecules and also 2 NADH molecules, reducing equivalents. So that's textbook knowledge. Now, the pyruvate that comes out of glycolysis can enter the mitochondria in the so-called Krebs cycle, which is coupled to oxidative phosphorylation. And this pyruvate is completely burnt to carbon dioxide and water. And that liberates more reducing equivalents, NADH and FADH, and also GTP. Now, in oxidative phosphorylation, the NADH and the FADH can be converted to approximately 3 ATP molecules, such that when you look at the overall yield of this, the Krebs cycle and the oxidative phosphorylation coupled to it yield about 16 ATP equivalents, whereas glycolysis only about 2 ATP equivalents. And actually, this is per pyruvate, this is per glucose. So in fact, it is even double. So that's what you probably have learned in your biochemistry classes at some point. and so this process, glycolysis, is pretty inefficient in terms of energy output per carbon input, whereas this one is pretty efficient. Now, what the biochemistry textbooks are sometimes a little bit, or not very clear about is that glycolysis is very fast, whereas oxidative phosphorylation is relatively slow. It's about 100 times slower than glycolysis. So now every textbook has it that glycolysis and the Krebs cycle are coupled to each other via pyruvate. Now, if one is much faster than the other, you probably all see that this is a problem. Because if you run glycolysis, you get all this pyruvate, and it cannot be processed fast enough, you run into a problem. You need to get rid of the pyruvate because that will feedback inhibit glycolysis, and then you're stuck. So this coupling is a problem. And the point that I would like to make today, or one of the points that I would like to make today, is that this coupling probably doesn't happen. Or at least not the way that it is presented in biochemistry textbooks currently. So one of the issues here is also that glycolysis happens in the cytoplasm, and the oxidative phosphorylation Krebs cycle happens in the mitochondria. So there has to be a transfer of molecules from one part of the cell to another. And I want to try and very briefly depict how this system evolved and what the problems with that are. So to understand how this evolved, we need to go back in time in the evolution of life on this planet. And what is shown here is a composition of the atmosphere in partial pressure. And we start when Earth formed about 4.5 billion years ago. And we look at what the atmospheric composition at the time was. Also we look at then when did life evolve? And the interesting feature, or to me at least amazing feature, is that the last universal common ancestor, Luca, lived on this planet already about 300 million years after the planet formed. So very early on. And Luca evolved and under atmospheric conditions that are very different from what we have today. Notably, the Earth's atmosphere at the time was rich in hydrogen. That hydrogen, in the primordial cloud that formed the solar system of planets, was a major component of the atmosphere. And when the sun ignited, eventually the solar wind blew most of the hydrogen away. But when life evolved, we had hydrogen. So we had reducing conditions. And under these conditions, glycolysis evolved. And we can actually see some biochemical traces for this, notably NADH, which is a hydrogen fast member, is probably the reason why it evolved this way is probably because there was a lot of hydrogen around. Now, a couple hundred million years later, oxygenic photosynthesis evolved. These are organisms that are related to today's cyanobacteria, and they release oxygen. And initially, that oxygen that they release doesn't really do much because there are a lot of things that can be oxidized, notably iron in the Earth's crust. So there are a lot of sinks that take away all the oxygen. So oxygen that is generated is not released into the atmosphere. But about 2.4 billion years ago, something happened, the so-called Great Oxygenation Event. And from that point on, oxygen rose in the atmosphere. And it rose to the levels that we have today, about 20%, eventually. So, so the Earth's atmosphere changed from a reducing atmosphere to an oxidizing atmosphere. And that changes completely how metabolism can be formed, or what kind of metabolism can be made. And notably, under these conditions, after the Great Oxygenation, oxidative phosphorylation evolved. Oxidative phosphorylation is actually basically oxygen photosynthesis turned the other way around. Okay, so then something happens a little bit later on, endosymbiosis and the evolution of eukaryotes. So this is where oxidative organisms that mastered oxidative phosphorylation, they joined with other organisms that mastered glycolysis and formed a unit. And these oxidative organisms, they later on became the mitochondria of the eukaryotes. So mitochondria are actually separate organisms, or originally separate organisms that joined symbiotically with other organisms that mastered glycolysis. So this, at this time point, about 1.8 billion years ago, this is where glycolysis and oxidative phosphorylation for the first time came together. They were kind of married together, but this marriage, as probably any marriage, was not totally, problem-free. And there are some breaking points when you marry these types of metabolism together that create problems even today. So, well, later on, when oxygen rose to levels, in today's levels, this gave rise to the evolution of multicellular organisms like us, for instance. Okay, so this unhappy marriage created some sort of problems, and these problems or these breaking points in energy metabolism, some of them were identified quite early. So Louis Pasteur in the 1850s already identified one. And at the time, he was contracted by the French winemaking industry. They wanted to make winemaking, alcohol production, they wanted to make that more efficient. So what he investigated, yeast in particular, and what he found was that when you have these yeast cultures and you expose them to a lot of oxygen, this promotes oxidative phosphorylation. Whereas if you have no oxygen, this drives metabolism towards glycolysis, which in yeast leads to alcohol production. So limiting oxygen drives on alcohol production. The winemaking industry in France was very happy about. Now, if we transfer this to the retina is very highly perfused, so there's a lot of oxygen around. And think about that. What should the retina use? Oxidative phosphorylation. Okay. Now, in 1923, Otto Warburg came along, and he studied metabolism, especially in the context of cancerogenesis. He was after understanding cancer. And what he did is he took rat tissues and put them in a Ringer solution and measured how much lactate they would release. And at the time, it was already known that lactate was kind of a surrogate for glycolytic metabolism. So a lot of lactate meant a lot of glycolysis. Now, what he found was the tumor tissue always releases a lot of lactate. So this was kind of his signature, tumor lactate production, whereas healthy tissue would not release lactate. And the only healthy tissue that he found in those days was paradoxically the retina. So the retina releases a lot of lactate, even when oxygen is present. So this he referred to as aerobic glycolysis, glycolysis in the presence of oxygen. And this is a clear contradiction to Pasteur, who said, if you have oxygen, you should do oxidative phosphorylation. Now, Warburg says, yes, everybody does it like this except the retina. So why is that? And then a couple of years later, Herbert Brace Crabtree, he took up those experiments. Actually, he kind of combined Pasteur and Warburg. And what he found was that when you have a high level of glucose, this drives the metabolism towards glycolysis, regardless of how much oxygen you have. So even if you have a lot of oxygen, if you have lots of glucose, the cells do glycolysis. So high glucose shuts down oxidative phosphorylation for some reason that we still don't understand to this day. Okay, so another point that we need to briefly introduce here is the idea of, or some ideas of how metabolism works in the central nervous system. And so I told you that the glycolysis and oxidative phosphorylation, they are chemically compatible. One delivers pyruvate and the other one breaks that down and oxidizes it. But they are kinetically incompatible. Glycolysis is far too rapid for oxidative phosphorylation to keep up. And that hasn't escaped people's attention. So it was basically already known since the 1960s. Now, to try and resolve this kind of kinetic incompatibility, Luc Pellerin and Pierre Magistretti came along with an idea that would basically split glycolysis and oxidative phosphorylation across two different cell types. They said, in the brain, we have glial cells and we have neurons. How about the glial cells doing the glycolysis and the neurons, the oxidative phosphorylation, and then this kinetic incompatibility, kind of result. The glial cells, they deliver pyruvate or lactate perhaps, and then the neurons use that and problem solved. Now, if we transfer this, this hypothesis, the so-called astrocyte neuron lactate shuttle hypothesis, to the retina, what would that mean? So in the retina, we have these RPE cells that are connected to the choroidal vasculature. So this is where stuff comes from, glucose notably. Glucose would come through the RPE cell, would enter the glial cell, a glial cell. The only glial cell that is close to the photoreceptors is the middle glial cell, so it should be that one. The middle glial cell then would split glucose into pyruvate and generate lactate from it. The lactate would be taken up by the neurons, the photoreceptors, and then they would use it for oxidative phosphorylation. That's an idea that one could have. Now, paradoxically, in 2017, the lab of Jim Hurley published a study, seminal study in eLife, where they found that the glucose that comes from the bloodstream through the RPE actually goes to the photoreceptors, and then the photoreceptors split it into pyruvate and lactate, and then the lactate is released to the RPE cell, and it would be the RPE cell that would do oxidative phosphorylation. So under this hypothesis, this is called metabolic ecosystem. Under this hypothesis, if that was correct, then the photoreceptors would do only glycolysis. That would be all they need. Now, the problem with that hypothesis is that photoreceptors in their inner segments are full of mitochondria. As you know, mitochondria do oxidative phosphorylation. And as a matter of fact, photoreceptors have the largest number of mitochondria of any cell in the human body or in any mammalian body. So if they have a lot of mitochondria, what are they using them for? Or in other words, if they did only glycolysis, they wouldn't need any mitochondria. So we know that in evolution, things that are not needed, we usually get rid of. But we still have them here, so they're probably used for something. So what's happening here? These are the questions that we're trying to resolve. And so one thing that we started doing about this is to look at the expression of glucose transporters. And we do here an immunostaining. These are retinal cross sections. Glucose transporter 1 here is stained in magenta. And as you can see, it labels the RPE cells. And there's a rhodopsin co-labeling, which labels the outer segments. And you can see there's a clear boundary, outer segments of photoreceptors and RPE cells, which have glucose transporter 1. Another glucose transporter that we have here is glucose transporter 3. And also here, there's a co-labeling with rhodopsin. And we see that glucose transporter 3 is expressed in the segments of photoreceptors. So that's an interesting arrangement, because glucose transporter 3 is actually a low affinity glucose transporter, whereas glucose transporter 1, low affinity, glucose transporter 3, high affinity. So if we have this arrangement, low affinity glucose transporter close to the bloodstream, where there's a lot of glucose, and then high affinity transporter in the photoreceptor segments, high affinity can take off the glucose of a low affinity transport. If you have, it the other way around, the flow of glucose wouldn't work. And this setup matches what we see also in the brain, where we have astrocytes that express glucose transporter 1, take out glucose from the bloodstream, and then the neurons express glucose transporter 3 to be able to effectively suck up glucose into their thalamus. Okay. So we looked at a number of different enzymes to investigate energy metabolism in the retina, and what is shown here are a couple of examples. RPE65 is a protein marker for retinal pigment epithelium, RPE. Glucose transporter 1 labels the RPE. Pyruvate kinase M2, this is strongly expressed in the segments of photoreceptors and the synapses of photoreceptors. Pyruvate kinase M2 catalyzes the last step of glycolysis. So this is a marker for glycolysis. And we have it in photoreceptors. Cytochrome oxidase, on the other hand, is a marker for oxidative phosphorylation. And we also have it in photoreceptor segments where we have a lot of mitochondria, and also in the synapses where there's also a couple of mitochondria. So judging from this, we would think that photoreceptors do both glycolysis because they have pyruvate kinase and oxidative phosphorylation because they have cytochrome oxidase. What we then do, we take mouse eyes, make retinal X-bond cultures from them, and cultivate them into on membranes, put them into 6-ball plates in this case, and then we can manipulate them any way we want under serum-free conditions, free of antibiotics, defined medium, and then we change this any way we want and analyze what happens. So one thing that we can do here, for instance, we can cultivate the retina with the RPE attached to it or without the RPE. We can inhibit glucose transporter 1 with a drug called 1,9-D-deoxyphosphate. We can inhibit pyruvate kinase M2 with a drug called chiconin, and we can basically abolish oxidative phosphorylation with a drug called FCCP. And the question is, if we manipulate these metabolisms, if we block glycolysis, if we block oxidative phosphorylation, what happens? And the first things that we looked at was what happens to cell survival. And to analyze this, we used the tunnel assay, label for dying cells, which is shown here in magenta. And we have these 5 different conditions: control, where we have very little cell death; a retina that is cultured without RPE, where we have more cell death. When we block glucose transporter 1 with 1,9-D-deoxyphosphate, we have more cell death. When we block pyruvate kinase M2 with chiconin, more cell death. And if we block oxidative phosphorylation, more cell death. And what's interesting is that the cell death basically happens exclusively in the photoreceptors. The rest of the retina, where we have the second-order neurons, in the inner nuclear layer, for instance, doesn't seem to care about this. So we can quantify the percentage of total positive cells in the outer nuclear layer, the photoreceptor layer, ONL. And under control conditions, there's very little. When we have no RPE, more glucose transporter block, more cell death, chiconin, and FCCP is really a killer. So this kills a lot of photoreceptors. Now, in the outer nuclear layer, you know that we have mostly raw photoreceptors in mouse, but also in human situation, and only about 3 to 5% cone photoreceptors. So what happens to the cone photoreceptors under these conditions? And here we use a cone arrestin-3 label to quantify all the cones that there are, and we count them, count the number of cones per retinal circumference per 100 micrometers of retinal circumference. And if we do this quantification, we see that under control conditions, we have a certain number of cones. If we take away the RPE, the cone number decreases, so that affects cones. If we block glucose import, blocking glucose transporter 1, that is really bad for cones because it basically kills them all. Blocking glycolysis, curiously, doesn't do so much, and blocking oxidative phosphorylation doesn't do anything to the cones. So the conclusion that we drew from this experiment was that the rods are really dependent on oxidative phosphorylation because they are very sensitive to FCCP. You know, if you have no cones dying here, all cones present, but lots of cell death, you know, what are the other cell type? What's the other cell type that's there? It's the rods. So these 20% dying cells, that must be the rods. So FCCP, ablation of oxidative phosphorylation, really is bad for rods, but not for cones. On the other hand, tampering with glycolysis, blocking glucose import, that's really bad for cones. So the conclusion was rods need oxidative phosphorylation. Cones appear to be dependent more on glycolysis. Okay, at this point we did we teamed up with Christoph Trautmann from the Werner Siemens Imaging Center and did a metabolomic study. where we analyzed well, hundreds of metabolites using nuclear magnetic resonance readout technique. And then there was some analysis going on that was done in this lab. And what they produced were these colorful labs for the 5 different experimental conditions and different metabolites that were significantly changed between these 5 conditions. And there's some unbiased clustering algorithm that then draws these colorful maps. And our initial expectation was that we would look at this, and then there would be patterns emerging, and then we would understand it all. That was the idea. And when you look at this, if you look a little bit closer, you'll probably understand that that expectation was far too, that hope was far too high. There are obviously things that are happening, and we can also do principal component analysis to see how these experimental groups are behaving. Are they really different? And the principal component analysis shown here suggests that really yes, there is something happening. They're really different, but what is happening? That's still a question. So, and there are other bioinformatics tools that you can apply to this, and basically that was all hopeless. Nothing worked. Nothing useful came out of this. So in the end, we ended up going through these metabolite lists, one metabolite after a time, at a time, and try and understand what each metabolite would potentially do or could tell us the changes in that metabolite. So, and one way of going about this is to look at markers for glycolysis. So I told you lactate, for instance, is a marker for glycolysis. Glucose splits, the glycolysis splits glucose into pyruvate, and if you have too much pyruvate, the cell can ferment into lactate. Okay, so lactate becomes a surrogate marker for glycolysis. And then we have the Krebs cycle and oxidative phosphorylation. And one metabolite that comes out of this that doesn't come out of anywhere else is GTP. At least under the conditions that we have here, GTP is a marker for oxidative phosphorylation. So what we can do now is we can build the ratio of GTP over lactate for the 5 different conditions, and that would indicate us, where's glycolysis happening, where's Krebs cycle and oxidative phosphorylation happening. So under the control conditions, there's a certain ratio. When we take away the RPE, that ratio goes down. And going down means there's more glycolysis happening. Or basically, because it's so low, it's basically exclusively glycolysis. On the other hand, when we block glucose import, the ratio goes more into Krebs cycle. Probably because the cells don't get enough glucose, and they need to produce energy somehow differently. And oxidative phosphorylation, being so much more efficient, is a good way of doing this. Blocking glycolysis with chicolate doesn't do really much about this ratio, but blocking oxidative phosphorylation really drives it towards glycolysis. Okay, so that's one way of analyzing such data sets. Can we do it better? Well, another thing that we can do is we can look at glucose consumption and lactate production. We can look at what is going not in the retina, but what is going on in the medium, the supernatant medium. How much glucose is consumed? So we look at these glucose concentrations, and these are not really, these are more arbitrary units. But in the control condition, we have a certain glucose consumption from the initial glucose level that we put into it. And when we have no RPE, there's more glucose consumption. Compatible with the idea there's more glycolysis. In the FCCP case, also less glucose, more glycolysis. And conversely, lactate production under the control conditions, there's a certain amount of lactate release. No RPE releases more lactate, more glycolysis, and especially FCCP releases even more lactate. Okay, so far we roughly understand what is going on. Under certain conditions, there's more glycolysis, and others, there's more oxidative phosphorylation. So now I want to look into what the RPE does to the retina. And I will basically focus on this situation for the rest of the talk. What is shown here in this pattern here is what happens when you compare a control retina that has an RPE with a retina that doesn't have an RPE. These are the metabolites that were found to be significantly changed when you compare these two conditions. Okay, so one thing that happens is that citrate levels go down. Now, citrate is a key metabolite in the Krebs cycle, in the tricarboxylic acid cycle or citrate cycle. And citrate is produced by an enzyme called citrate synthase. And here we have an immunostaining for citrate synthase in the cross-section of the retina. And you can see that there is citrate synthase expression in the intranuclear layer and ganglion cell layer, but there's in particular in the photoreceptor segments and in the synapses. So if there is a change in citrate here, it probably comes from photoreceptors. It could potentially also come from ganglion cells or so. Another thing is lactate. So lactate under no RPE, in the no RPE condition, strongly accumulates in the retina. And the enzyme that produces it is lactate dehydrogenase A, here shown in magenta, and that is basically exclusively expressed in photoreceptors. So these changes in lactate have to come from the photoreceptors. And then we have here alanine, which is also interesting. And the enzyme that produces alanine is alanine transaminase, or ALT. And what we have here is a co-labeling of ALT with cone-arrestin. And you can see that in the outer nuclear layer, alanine transaminase is expressed only by cones in the cone segments. So alanine, something happening in the cones. That's what we could conclude here. Now, other interesting metabolites are leucine, isoleucine, and valine. These are so-called branch chain amino acids, and for some reason they're accumulating when you have no RPE, but not under normal conditions. And another thing that is really interesting is that we have this lactate accumulation that Otto Warburg found only when there's no RPE. And when you go back to his 1923 papers, you understand that what he did with the retina is to explant it without the RPE. So what he measured, the lactate release that he measured, is probably an artifact from not having RPE. At least that's what I'm proposing here. So in normal conditions with an RPE, the retina wouldn't do it. Okay, or at least not so much. Okay, so now the alanine. Why is the alanine changing here? To try and understand that, we need to go also a little bit back into biochemistry, basic general knowledge. And we need to talk about Cori cycles and Cahill cycles. Now, Cori cycle, this was discovered in the 1930s by a biochemist couple. They got the Nobel Prize for it later on. So the Cori cycle has it that glucose in the bloodstream enters muscles. We're talking about muscle here in this case. Glucose is split into pyruvate that releases NADH and ATP. And then the pyruvate is reduced using up the NADH that was just generated to lactate. The lactate goes to the bloodstream and then enters the liver. And in the liver, the lactate is used to regenerate pyruvate. And then is going into gluconeogenesis, producing glucose. The glucose goes to the bloodstream, closing the cycle. That's the Cori cycle. Okay, discovered 1930s. Now, and every biochemistry textbook has it. Every medical textbook has it. Now, another cycle that is probably far more important was discovered later on, the so-called Cahill cycle, which has it that glucose from the bloodstream goes to the muscle. Okay, it's split into pyruvate. That's still the same. But then the NADH isn't used to reduce the pyruvate. Instead, pyruvate is transaminated with the amino group from glutamate that generates alanine, and the glutamate without the amino group that delivers alpha ketoglutarate. Okay? Now the alanine goes back to the bloodstream. In the liver, it is deaminated. Then you get pyruvate. The pyruvate is used for gluconeogenesis, closing the loop again. That's the Cahill cycle. Okay. So the interesting thing is, if we can measure lactate and alanine, if we see them, we know which cycle is going on. So in one case, a lactate, in one case, alanine. So we can use this as signatures. And now, what kind of metabolism could we do with this? So if we suppose the Cori cycle is what is happening here, the Cori cycle would have a glucose split into pyruvate, is reduced to lactate. Now lactate is released and taken away. Okay, that's Cori. And the interesting thing is that the Cori cycle doesn't need the Krebs cycle. So we can run glycolysis totally independent of the mitochondria of oxidative phosphorylation. And so we uncouple glycolysis and Krebs cycle from each other. Another possibility is to use the Cahill cycle, and that is far more interesting from an energetic perspective. And I will show you this. So in the Cahill cycle, we don't rely on glucose, we rely on glutamate. The glutamate, together with pyruvate, is transaminated, generating alanine, pyruvate plus amino group alanine, glutamate minus amino group alpha ketoglutarate. Now, the alpha ketoglutarate happens to be a metabolite of the Krebs cycle. It can enter the Krebs cycle. It can be further processed to oxaloacetate through various steps, and that frees NADH and GTP. And now, if you do this all the time, you keep on producing more and more oxaloacetate, you need to get rid of the oxaloacetate. And one way of doing this is using the enzyme pyruvate carboxykinase that generates pyruvate from this, and that kind of closes the loop again. So here you would run the cycle on glutamate input and alanine output, generating far more ATP than you did with the Cori cycle. Okay, this is independent of glycolysis. So you can separate these things. We could run Cori cycle and Cahill cycle in parallel without them interfering with each other. Now, the interesting aspect of this is that this what is here, the upper part of the Krebs cycle, this also contains the rate limiting steps. The first steps of the Krebs cycle are the rate limiting, really slow steps. If you don't need them because you start the Krebs cycle later, the whole thing runs a lot faster. So this kinetic problem that you have with the Krebs cycle kind of resolves itself. Now this lower part of the cycle is about 5 times faster than the full cycle. So it's not only delivering more energy, it's also faster, delivering more energy per time. Okay, now I told you about this enzyme called pyruvate carboxykinase, that you would really need to do this. If you don't have it, this would work. Now, where's that enzyme expressed? So we have 2 isoforms, the cytoplasmic, PCK1, which is strongly expressed in photoreceptors, and PCK2, which is strongly expressed and exclusively expressed in the retina in cone intersegments. So in cones, this would run really well. So if we look at this comparison, metabolite map, control retina with RPE versus a retina without RPE again. And there are other metabolites that are changing. So I was talking about alanine and others. Another interesting metabolite is aspartate. Okay, aspartate is really low when there's no RPE. Why is that? Another metabolite here is glutamate. And glutamate and aspartate, they behave exactly reciprocal. So what's behind that? Yet another metabolite that I would like to draw your attention to is N-acetyl aspartate. N-acetyl aspartate, so acetylated aspartate, behaves like aspartate. So what are these metabolites? Why are they behaving this way? And what's the enzymes that would make them, converge into each other, or that could convert them into each other? And the enzyme that's shown here, this is aspartate aminotransferase. Some people also refer to this as glutamic oxalic acid transferase, but we refer to it as aspartate aminotransferase. And this enzyme is strongly expressed in the retina, the AST1 isoform, and the AST2, that's the mitochondrial isoform, is expressed also. You can see this line up there in the inner segments of photoreceptors. So photoreceptors would be able to convert glutamate into aspartate by transaminating this. And what would that do to metabolism? And going back to this, going back to this diagram, one thing that came out in 23 was that this aspartate aminotransferase is actually important for photoreceptor survival. So this was published by the group of Thomas Rubin in Michigan, and they call this enzyme GOT1, and there's actually other names for it in the literature, which kind of adds to the confusion, because if you think this is called like this, other people call it differently, and that may cause confusion, but this is the same enzyme. GOT1 is AST1, and they found that when you ablate that in photoreceptors, or rock photoreceptors in particular, they die. So it's obviously important. So how could it be important? Now, if we look at glycolysis in Krebs-Cycle again, we've already seen with the Cahill cycle that the Krebs cycle can be abbreviated. Can we do this also more directly? And if we look at this, it's possible. We can take glutamate and aspartate aminotransferase can convert glutamate into aspartate, taking off the amino group. That would yield alpha-ketoglutarate, same as in the Cahill cycle. And if we transfer the amino group from glutamate to oxaloacetate, we get aspartate and alpha-ketoglutarate. Okay. Now, of course, if you keep on adding glutamate to it, you would end up having a lot of aspartate. And aspartate is not membrane permeable. You need to get rid of it, because otherwise it will fog up the works. So, how do we do this? Well, first of all, we can run the Krebs cycle, this lower part of the Krebs cycle, with alpha-ketoglutarate, delivering GTP and NADH and KDP equivalents. But we need to get rid of the aspartate. So we can assimilate it with acetyl-CoA, and acetyl-aspartate is membrane permeable, and then we can just get rid of it. So if this was happening, what we would have is glutamate input and acetyl-aspartate output. And if we ever had a problem with not having enough acetyl-CoA, we can use branch chain amino acids that are depleted in the normal retina to generate acetyl-CoA. And if that wasn't enough, well, we can use pyruvate carboxykinase to regenerate pyruvate, and from pyruvate we can make acetyl-CoA. So we have kind of two loops intertwined, where basically input is glutamate and output is acetyl-aspartate. And again, we skip the upper half of the cycle, the rate limiting slow steps, making the whole thing faster. So this Mini-Krebs cycle that was first suggested in the 1990s would be efficient. It would be independent of glycolysis, uncoupled from glycolysis, and it would be relatively fast. Okay. So what I've tried to show you today that the retina may have access to 4 different pathways for energy production in parallel. First of all, the conventional biochemistry textbook cycle, where we have glucose split into pyruvate entering the Krebs cycle and being burned completely to carbon dioxide. The Cori cycle, where the pyruvate is not entering the cycle but being reduced to lactate. The Cahill cycle, where we use glutamate input to generate alpha-ketoglutarate and alanine, and the Mini-Krebs cycle, where we use glutamate and to some extent branch chain amino acids to generate energy and acetyl-aspartate. Okay, so we have 4 different pathways here, and now we should briefly look at how much energy does this deliver, and why it could one or the other be interesting under certain conditions. So if we look at glycolysis, the only glycolysis, per pyruvate molecule, what we get in the cytoplasm is 1 NADH molecule and 1 ATP molecule. Well, that's not so much. If we couple it to the Krebs cycle, using oxygen, obviously, then of course we get a lot more NADH and FADH equivalents and GTP, making the whole thing very efficient, but slow. The Cori cycle, here we would use up the NADH that we generate in glycolysis to reduce pyruvate to lactate. So we are what remains is only 1 ATP. So nothing much. In the Cahill cycle, where we have to kind of well, if we assume that is happening only in the mitochondria, that the cytoplasmic steps are not delivering any energy, then we're remaining with 3 reducing equivalents that can be converted each into 3 ATP and 1 GTP, so 10 ATP equivalents. And then the Mini-Krebs cycle, we would end up, if we run it on glutamate and branched chain amino acid input, we would eventually come up with 6 reducing equivalents. 6 times 3, that's 18 ATP equivalents plus 2 GTP, that's 20. So that's very good. And it's fast. So that's interesting. So then the next thing that we need to look at is the rate of energy production. So, and you may imagine that for a photoreceptor or for a cell neuron, it's not only important to be very efficient in terms of ATP output per carbon input, you may also need your ATP at a certain point in time. Now, if you have if you are a neuron, if you were a neuron, you just fired an action potential, you need to repolarize your membranes fast. You need a lot of ATP for that. And if you don't get it, you die. So the rate is important. And now if we think about glycolysis, so as it would happen in the Cori cycle, and we just say there's 1 ATP unit that comes out of this per pyruvate input, and then there's a certain unit and 1 unit of time that is used to this. And we compare it to the conventional Krebs cycle, where pyruvate is oxidized to carbon dioxide. This would be about 100 times slower. The yield would be higher, but it would be much slower. If we compare it to the Cahill cycle, it is not as efficient as the Krebs cycle in terms of ATP output per carbon input, but it is faster. So the Cahill cycle, from a kinetic point of view, is far more interesting. And the Mini-Krebs cycle is even more interesting. And if we look at this 45-degree angle ordinate, which would indicate same ATP production per time as in glycolysis, you can see that the Krebs cycle is far less efficient per time, and the Mini-Krebs cycle is more efficient per time. Okay, so this brings me to the conclusion. So what I would like to summarize is that photoreceptors, they probably use not only glucose as fuel to drive the metabolism, but also glutamate and branched chain amino acids. Rods apparently are quite dependent on oxidative phosphorylation. Cones, they definitely need glycolysis. They're less dependent on oxidative phosphorylation. That doesn't mean that they don't do oxidative phosphorylation, but they can live doing only glycolysis. Rods, from the data that we have so far, seem to be using the Mini-Krebs cycle. Should be a little bit careful about this. It could be also in cones. Cones are more likely to use the Cahill cycle, and the Cori cycle it could also use. A key point is perhaps that lactate, alanine, and N-acetyl aspartate, these are signature metabolites, so such that when you see them, you know what kind of metabolic pathway is running, under what condition. My former PhD student, Yi Chen, she tried to put this into some sort of graphical abstract, which would have that, the rod photoreceptor on the left would use Mini-Krebs cycle and perhaps Krebs cycle, whereas the cone photoreceptor would be using the Cori cycle and Cahill cycle, and this would be driven by glutamate and glucose input, releasing to the top towards the bloodstream, N-acetyl aspartate, lactate, and alanine. Now, N-acetyl aspartate, just as a fun fact, is one of the most abundant metabolites in the retina and in the brain. Okay, so the idea is that we use these different metabolic pathways to match the yield and the rate of energy production to the current metabolic needs. And we know that photoreceptors when they switch from darkness to light, their energy consumption, their energy demand changes dramatically on a millisecond timescale. So this is probably one of the reasons why they need to have access to different metabolic pathways to really, produce energy when they need it. Of course, there are a number of other questions. So what are the kinetics of the different pathways? We have rough ideas, but we don't understand it really fully. Can we do modeling of this to try and understand this better? And if we have these different energy production pathways, and it looks that rods and cones are really different here, would that explain the differential vulnerability of rods and cones that we see in certain diseases, such as retinitis pigmentosa or achromatopsia or AMD? And then, the Crabtree effect, what is the molecular basis for it? To this day, it was discovered 1929, it's a long time ago. We still don't know how it works. And of course, if we eventually understand this properly, could we use it to rationally design novel therapeutic interventions? That's the final goal, basically. Okay, so finally, I would like to acknowledge people that were involved in this work. Of course, our cooperation partners. There are some publications that came out of this work. Of course, we'd like to thank our funding, our funders. And finally, I would like to point out that this work basically gave rise to a new doctoral training network we call Limits to Vision between France and Germany, between the Institut de la Vision in Paris and the Institute of Ophthalmic Research here in Tübingen. And this new doctoral training network will likely start in January next year. So you may see some more PhD students working on questions related to this in the future. That's it from my side. And if there were any questions, I'd be happy to try and address them. Yes, Angie.
First of all, beautiful presentation. Very, very nice.
Thank you.
I like it. I have many questions, and I will try to use it to talk about the others in another moment. What about glutamine? So the glutamine, because the blood we should be glutamine, are the RP transformed in glutamate and giving to the photoreceptors? Can it be? If you check for glutamine synthetase or glutaminase?
Okay, I'll try and draw this very quickly. So I was actually yesterday, I was working on doing on this talk, and I was thinking whether I should talk about glutamine, because the data that you see in the paper actually has interesting aspects of glutamine. So if we have a photoreceptor, let's say this was a rock photoreceptor, cell body synapse, and then we have a Müller cell. All this process is in wrapping the photoreceptors. So the neurotransmitter of photoreceptors is glutamate. Okay, the glutamate is released at the synapse, and then the Müller cells, one of the key markers of Müller cells is glutamate aspartate transporter, GLAS, which takes up the glutamate from the synaptic cleft and puts it into the Müller cell and then transforms it, and that's the next step, into glutamine. And the enzyme for that is glutamine synthase, which is another key marker for Müller cells. So now the glutamine, one of the ideas is that the glutamine goes back into the photoreceptor synapse, the photoreceptor then splits off an amino group, gets glutamate, kind of closing the loop that there is always sufficient glutamate as neurotransmitter. Now, what we found is that the enzyme that splits glutamine in photoreceptors is strongly expressed in the inner segments of photoreceptors. This is glutaminase C. So, and the transporter for glutamine is strongly expressed in Müller glial cells, all along. So that would imply that the glutamine can not only go into the synapse, but that it could also go into the inner segments, and then it could be used as converted to glutamate and used as fuel for mitochondria. And there's a very recent paper that came out by our competitors, Thomas Gubin in Michigan. I think last month or so, they published a paper in eLife where they showed that a knockout of glutaminase C, GAC, a knockout of glutaminase C kills photoreceptors, raw photoreceptors in particular, implying that this is a relevant pathway for photoreceptors, for photoreceptor viability, likely for photoreceptor energy metabolism. The exact details are still.
Because I also remember that from the cancer research with glutamine, glutamate, or aspartate, they are also physically separated in the cell, in the mitochondria and the cytoplasm that would fit also to the separation of the compound. I don't remember which one is produced in the mitochondria and then which one is active in the cytoplasm of the circle of glutamine, glutamate, aspartate, asparagine.
Yeah.
But maybe it's important.
Yeah, when it comes to the subcellular resolution of this, we're still not very, very good because is this enzyme expressed in the mitochondria in the inner segments, or is it expressed in the cytoplasmic space around mitochondria? I couldn't really tell you. The resolution of what we have used so far, immunostaining is not good enough.
And the RPE with glutamine, glutamate, do you know anything?
I can't really tell you. What I can tell you, so when you look at this map, comparison of metabolism with a retina with RPE, retina without RPE, glutamine was not significantly changed there. So that could imply, but I'm not saying that it does. It could imply that glutamine is not so important for the RPE photoreceptor crosstalk. But it may also be that the resolution of our method is not high enough. Are there other questions? Yes.
Can't you imagine being the setup where you use explants from mice that have only rods or only cones to really check out?
Yeah, that was a suggestion from one of the reviewers as well. And yes, so. We have these mouse models for achromatopsia, like CPF1 mouse, which at a certain point have only rods because the cones die because of the mutation. So rod only is relatively easy. Cone only, there's this CRX mouse where the differentiation, transdifferentiation from cones to rods doesn't really work, and we get only cones or photoreceptors that are a little bit like cones. So we could use that as a converse example. That was also the reviewer's suggestion. Yeah, it's a lot of work. We haven't done it yet.
But then in that case, if they are in an ecosystem altogether, if you have only one type, it would adapt to availability of a substrate that the other cell is not using in that time. It's the same point of it without RPE. I mean, we of course are using some.
For sure, the analysis of this, the experiment as such, is probably not so complicated, but the analysis of this would be pretty tough.
Have you tried them?
Which one?
Tried blend, so like in the gym with
No.
Because you could then identify between my rods and cones because it defines between the two processes. And you don't have to add any additional staining.
Yeah, so no, we haven't tried this, and mostly because we don't really have the technique established here. But what we're now thinking of doing, and this is again a collaboration with the lab of Christoph Pottwein, is to use MALDI imaging to image the metabolites in retinal cross section in individual cells directly. Problem of the method is spatial resolution and sensitivity. We're trying to set it up, and actually trying to get that to work. I hope it will work. Potentially, it could deliver a lot of interesting data, but we'll have to see how well it actually works.
Because it would be quite interesting to try it that would help define between the two receptors if they're working on the two different pathways. One's more glycolysis and one's more on spots, so you should be able to see the distribution and then also look at the effect between the RP and the photoreceptors.
Yeah, and my understanding is that with film, the resolution of individual metabolites is not so easy.
It's not necessarily metabolites, so you're basing it on FADH and NADH, and it's the ratios between the two.
Yeah.
Because of various mutation and wavelength. But it can really help with defining different areas and switching between metabolites. So if it's switching from one process to the other, they use it quite a lot with cancer, obviously with cancer, but also to look at treatments and penetration of drugs into microtumors. So you can see if it's really affecting or actually generating an effect. So it can be quite sensitive, and it can be for multiple cell layers as well.
Yeah, I guess at some point we'll have to look at the different methods to try and see which one works best, or whether they're confirming each other.
Yeah, I think it would be with combination of something else. It won't be on its own. But could be useful too.
Yeah, I agree. Josie?
Yeah, maybe a stupid question, but is it a coincidence that the amino acids they use as a fuel are the essential amino acids? So the body cannot produce them, or is it?
Probably not, but you mean glutamate or branched chain amino acids?
Yes.
Yeah. Yeah, I would say it's probably not a coincidence, but yeah.
Okay, and I have another question. Do you check for other tissues in the body where something similar happened, or is there something known about other tissues or cancer tissue where it's?
Yeah, so in cancer tissue, in some ways behaves like the retina. Notably this, lactate release that I talked about already. They also seem to be using branched chain amino acids a lot, or tumor cells in general, although tumors can be very, very different one from another. But we haven't looked into this. So we haven't, we're not cancer people.
No,